Benediktas Valys from the University of Sheffield, who's going to talk about HMM-based clustering of multimorbidity trajectories in one of our contributed talks.
So I'd like to present my PhD project. So I'll talk about a two-stage hidden Markov multimorbidity clustering pipeline. So it's a bit of a mouthful, but we'll start with the background. So what is multimorbidity? So multimorbidity is the presence of two or more chronic conditions in the same individual. So multimorbidity is quite prevalent in the elderly population, but it still remains poorly understood. So it's still unclear why certain conditions co-occur in some populations. And to investigate this, we cluster temporal patient sequences. So at the moment in the literature, the most clustering of multimorbidity patients is done using static data. However, our goal is to cluster the temporal patient sequences because multimorbidity involves over a long period of time, and this allows us to better investigate how this evolution occurs. So to tackle this, we propose a two-stage pipeline. So the first stage involves clustering conditions, and the second stage involves clustering patients. So the data that we work with is composed of binary vectors. So this is an example of time point 1 from patient 1. So the green indicates that patient 1 at time point 1 was diagnosed with condition 1. And then over the course of the time that the patient was observed, in time point 2, he was diagnosed with 2 more conditions, time point 3, even more conditions, and this accumulates over the entire observation period. So the entire data set is composed of n total subjects, and each of those subjects is described by a sequence of these binary vectors. So in the first stage of our pipeline, we cluster conditions. So we first apply multiple correspondence analysis to reduce the dimensionality of the data set and to transform the values from binary to continuous. Next, we cluster the time points using k-means, and these clusters of time points we call characteristic disease groups. So these characteristic disease groups contain commonly co-occurring conditions. So this is an example of basically what so all of the time points of the entire data set are grouped into individual CDGs, so characteristic disease groups. So they're colored in different colors. So this allows us to then transform the data set from the original binary vectors. So time point 1 of patient 1 goes into CDG1. Time point 2 goes into the second characteristic disease group, and then the rest follow. So we do this for all the subjects in our data set. So all the subjects get transformed from the binary into the sequence of CDGs. And next, we train a hidden Markov model using these sequences of characteristic disease groups. Afterwards, we take each sequence of these characteristic disease groups and then pass them through the HMM using the forward-backward algorithm to produce an induced transition matrix. So we apply this same process for, again, the entire data set, and we find all the induced transition matrices of all the subjects. We then compare all these transition matrices using the Bhattacharya distance measure and produce an m by n distance matrix. So this distance matrix is then fed into the dbScan clustering algorithm, and we finally produce subject clusters. So each subject is placed into a separate cluster. So this is the entire pipeline in detail, and the 2 stages are separated out. So the whole pipeline contains a number of parameters that need to be pre-specified. So the number of dimensions in the MCA transformation, then the number of k-means clusters, the number of hidden states, and then 2 parameters for the dbScan clustering. So to decide on the specific set of parameters, we use Bayesian hyperparameter optimization, and we aim to maximize the silhouette indices of the subjects and the condition clusters. So the silhouette index is a measure of clustering quality. So a high silhouette index means that clusters are well separated and they basically represent distinct groups of data. So a high silhouette index is good, and we aim to get high silhouette indices in both our clusterings. And the Bayesian hyperparameter algorithm that we use is the tree of Parzin estimators. So to analyze, we chose the SNAC-K dataset, which is a longitudinal population-based study from Sweden. It's a study of over 3,000 adults above the age of 60. Each subject had a maximum of 5 observations over a 12-year period. And so after running the hyperparameter optimization, we found that the number of characteristic disease groups and the number of subject clusters was commonly around 3 to 16. I can go into more detail about this because it doesn't seem that reassuring. So I can talk about this afterwards. So more results. So these characteristic disease groups, we named them after the condition with the highest disease group score, which essentially measures how prevalent a specific condition is in a specific disease group. So out of the clusters of those time points, which conditions were most unique to a specific disease group. So during our optimization, there were a number of disease groups that co-occurred. So they weren't just split randomly into just a complete mess. There were several that co-occurred throughout the many runs. So we had eye conditions were commonly grouped together, then heart conditions, lung, musculoskeletal conditions were found in distinct disease groups. So we also worked with a group of expert clinicians to validate our results, to make sure that they were useful and could be used in further research. So this is an example of the final results. So this is, these are results of the disease group clusters. So these are clusters of conditions, and these are 4 disease groups. And at the top of the table, they're named after essentially the theme of the disease group. So there's the general one, which contains obesity, dyslipidemia, etc. And we have the heart conditions with a number of heart conditions. And the CDG size is the number of time points in a specific disease group cluster. So, and the results of the second stage of the pipeline are the final subject clusters. And this diagram basically represents how this specific cluster of 590 subjects transitions between the entire observation period. So how it transitions between these disease groups. So this is another example of a different cluster with more disease groups. So there's the previously mentioned eye conditions, and again, the transitions between the 5 time points. Excellent. Yes, so we found that optimizing the silhouette index of the characteristic disease groups and the subject clusters simultaneously provided more interpretable results. So we tested the pipeline by first splitting the optimization and first maximizing the silhouette index of the disease groups. But we found that doing the optimization simultaneously produced more understandable and interpretable results. So, and further work includes testing the pipeline with alternative algorithms, then conducting a wider parameter search, and then dealing with the sparsity of the data set as well. Thank you to everyone, and I'll take some questions.